Hello, my name's Connor West, and I'm a second year PhD student at the Medical University of South Carolina. And today I'm going to talk to you a little bit about my research entitled Sugarcoating the Bad News Cells Do It Too. So, I think we can all relate to a time that we had to do some sugarcoating of bad news to not make the news seem so bad. But did you know that cells also like to sugarcoat? On each cell surface, we have a complex mixture of sugars called glycans. And when a cell turns cancerous, this cancer cell forces these glycans to change and better help the cancer cell adapt and grow in the body. My research focuses on how exactly we can tell these changes earlier on and to hopefully stop the cancer before it becomes a problem. To help explain this a little better, I want you to think of an M&M for a second. I want you to think of that yummy candy coating on the outside around a chocolatey center. Now, imagine that's your cell, where the chocolate is the inside of the cell and the candy coating is the sugar coating of glycans. Now, imagine for a second that normal cells are blue, green, yellow, and red M&Ms, and they're happy and healthy, functioning inside your body, nothing's going wrong. But now, imagine one becomes cancerous. That cell surface is now going to appear like a brown M&M, which makes it easy to see where the cancer is in your body. Now, obviously, that's a little bit of an oversimplification of what happens, but the principles are essentially the same. So, what we're trying to do specifically is we're trying to find these brown cells in your body and be able to find them earlier, faster, and better. To do this, we use a tool called mass spectrometry. Now, again, imagine the M&Ms. I want you to think that each color of the candy coating has a unique weight, like 1 gram for a blue candy coating, 2 grams for a yellow candy coating, and 5 grams for a brown candy coating, etc. And what we do with mass spectrometry is we take the cells and break off the candy coating, leaving just the chocolatey center, and then we weigh the candy coating itself on a super sensitive scale. And if we know the weight of the candy coating of cancer cells, we can identify which cells were cancerous by the weight. It's a pretty cool technique, but it does have its own inherent struggles. And while I wish it was as easy as I just described it, it unfortunately doesn't work that way. In following the previous analogy, most cells aren't just one color, they're a mixture of colors, and the body doesn't just have 5 colors, but hundreds of different colors. So the difficulty comes from finding out exactly which color or pattern of colors is cancerous, finding out how much that one specific color or pattern weighs, and then finding ways to identify that color without having to break the sugar shell off of your cells. For example, we can develop drugs that target specific colors or combinations of colors to help therapeutically treat cancer cells, or we can find out what's causing the colors to change in the first place and use a drug to target whatever is forcing that color change, which is what my project focused on specifically in liver cancer. So, I hope you were able to learn a little bit about these sugars or glycans and how they change in cancer progression, and how we can study those changes using mass spectrometry to better determine exactly what makes a cancer cell cancerous and create better medicines and diagnostic tools in response. Thanks for your time and attention, and I would be happy to answer any questions you may have about my research, and I sincerely hope that I didn't ruin brown M&Ms for the rest of your life. Have a great day, and I hope to hear from you soon.